Good morning, everybody, and welcome to the annual Gairdner High School lecture hosted by none other than the Collegiate at the University of Winnipeg. A special, round of applause. There we go. And a special virtual welcome for those that are watching in attendance online as well. We acknowledge that we are gathered on ancestral lands on Treaty 1 territory. These lands are the heartland of the Métis people. We acknowledge that our water is sourced from Shoal Lake 40 First Nation. The Gairdner Foundation was established in 1957 with the main goal of recognizing and rewarding international excellence in fundamental research that impacts human health. The Gairdner High School lecture has been hosted by the Collegiate since when? Anybody want to take a guess? Close, 2015. And it's nice to come back in person. And what better place to have it than at a high school at a university? Here, where we have, here's our little plug-in, high quality, flexible secondary school education with small class sizes, amazing instructors, handsome associate deans, and no, I'm joking. And that only equips our graduates for lifelong success. Now, the Gairdner Foundation has presented over 400 awards to laureates from 40 countries, and 96 of those have gone on to receive the Nobel Prize. Today, we are fortunate to have a professor in the Department of Biochemistry and Molecular Biology from the University of British Columbia. I would like to highlight a few of the speaker's long list of accomplishments, because there's so many, and you might have seen it on the poster advertised. But in his spare time, he likes to co-found or begin 11 biotech companies, publish more than 350 scientific articles, and is an inventor of over 60 patents. Now, as the pandemic took over our lives, our presenter was busy working with a team to develop a COVID-19 mRNA vaccine. So to tell us more about the story of the mRNA vaccine and why research is interesting, please join me in welcoming the Canadian Gairdner International Award recipient and also a member of the Order of Canada, Dr. Pieter Cullis.
Okay, this is, I haven't really given a talk to high school students before, so I've had to adjust some of the contents from what I would normally be presenting. So I thought I'd start off with a little bit of many of you probably don't know why research might be interesting or why it might be something that one would want to do for an awfully long time, such as I've done. And also what some of the ways that research moves, it's a very non-linear process. There's things that connects you wouldn't normally associate with that. With say the stories of the COVID-19 vaccines, we never started to really go in that direction at all. It just ended up that we did something that turned out to be important. And as indicated, I've started a number of companies. And some of those have been very much involved with the work that we've done. So let's go back in my history. I graduated in 1962, so that's 60 from high school. That was 60 years ago. Probably makes me completely irrelevant to anything that might be of concern to you. I attended high school in West Vancouver. And my father taught physics in that high school, so it seemed only natural to go on in physics. And then this is in UBC, University of British Columbia. And so then I ended up with a PhD in physics in 1972, and then I decided that I couldn't stand physics anymore. It's a very well established, anyway, there was a number of reasons. It was very difficult to discover anything new. Also, the social life wasn't exactly that brilliant either. So in 1973, I won a scholarship to go to Oxford to study biochemistry, and I didn't know anything about biochemistry, but I thought, okay, this could be interesting. I was using a physics technique, so I did have some sort of entree. Now, in that process, I got interested in something I'll try to tell you a little bit about, which are membrane lipids. And then since then, using lipid-based systems to deliver, well, cancer drugs for cancer chemotherapy, and also a messenger RNA, and I'll explain what those things are, siRNA, small interfering RNA. I've been doing it ever since about the middle 80s, early 80s. So I've been a professor in the UBC Biochemistry and Molecular Biology Department since about 1978, so I've been there for a long time. But I've been doing, as was indicated, we started a bunch of companies that now employ about 400 people in the Vancouver area. So why is life science research interesting, or why have I found it interesting? And really, it's a matter of you're trying to understand how nature does stuff. And what role all of the components in a biological system, what role they play. And so I got interested in biological membranes, and there's lipids in biological membranes that play particular roles, structural roles. As is sort of indicated on the right-hand side here, these are lipids that encapsulate. In this case, we're saying it's an anti-cancer drug or anything else you might want to encapsulate. And again, there's lipids here surrounding a piece of RNA or DNA. And so what we found was that we could use all this understanding of membrane lipids, that was really basic stuff, to deliver these cancer drugs and nucleic acid drugs. So we've got 5 drugs approved now by the FDA, the U.S. Drug Administration, as well as the European Medical Agency. So they're being used to treat cancer and some of its complications. And of course, 2 drugs, they're using the nucleic acid-based drugs, and one of those is Comirnaty. How many people in the audience have had the Pfizer vaccine? Yeah, so those of you you've had the vaccine that we ended up inventing. It's the fatty the lipid nanoparticle that surrounds the messenger RNA in the vaccine. So why are lipids in biological membranes interesting? Well, in your body, you've got 30 trillion cells, right? It's a lot of cells. They make up everything in your body, from your bone to sperm to egg to muscle, et cetera, et cetera. They all have different morphologies, but they all have one common thing, and that is they're all surrounded by a membrane. And so this is the membrane around them. And of course, inside a cell, as you probably learned in biology, you've got all sorts of other things called organelles, and each of those organelles has a membrane around it too. And so the membranes are kind of important. They allow biological processes to be separated from an inside and an outside. And so you can do very specialized things inside these membranes. So they all rely on lipids. So this is a biological membrane. It's got proteins that do all kinds of things that allow various ions to get across, or they transport whatever. But they all rely on this basic structure, which is a lipid bilayer. And so this is made up of what's termed the membrane lipid, which has one end that likes to be near water and the other end that likes to be sequestered away from water. So they're quite simple molecules in a way, but they certainly are vital to anything you do. The membranes are pretty darn important. So membrane lipids, for a long time, this just happens in science, they're not very, popular. The people would say, okay, well, what we really want to study are the membrane proteins, because they do all the work. But we stuck with, well, we want to understand what these lipids are doing, and it turned out to be actually pretty useful. So really basic things we got interested in, there's lots of lipids in membranes. They don't adopt a bilayer, they adopt these other structures. Well, what are they there for, and what are they doing? And and also the lipid composition on one side of them, I mean, just to make things extra complicated, there's hundreds of different lipid species, but there's different composition on one side compared to the other. And so what drives that asymmetry, and what functional roles does it play? I mean, as you can tell, this doesn't have much to do with the vaccine, but it actually turned out to be quite important to our development of a vaccine later on. We found that the lipids that don't like to be in a bilayer normally, they play a big role in membrane fusion. So that's pretty vital to life, fertilization, and various other forms of self-transport. So it was one kind of interesting ramification. We also found that we could drive asymmetry by just having a pH gradient across. I think most would be familiar with what a pH gradient is. So the outside is about pH 7, which is a normal physiological pH in your body. pH 4 is about the acidity of a lemon. It's not very acid. But what we developed was this ionizable cationic lipid that had the property that at low pH, it became protonated. A proton would associate with the head group, so therefore it was positively charged, whereas at neutral pH, the proton would dissociate. And so this ended up driving lipid asymmetry because the neutral form was membrane permeable. It would get inside, get protonated, couldn't get back out because these lipid bilayers are very impermeable to charged substances. And so we could generate lipid asymmetry. So why is this important? Well, it turns out that DODAP, these ionizable lipids, are absolutely vital to the vaccine that a large number of you have had administered to you. But what we found as this was in the middle 80s, we found that we could take these vesicles and we could load cancer drugs into them using a pH gradient approach. So this is doxorubicin. It's a very common anti-cancer drug. And what we found was we could load, say, 100,000 of those into one of these little vesicles that's 100 nanometers in diameter. Now, 100 nanometers is pretty small. I mean, it's about 1/100th the size of a cell. A cell is, if you draw a line on a piece of paper, you can fit 10 cells into the width of the pencil line. So you can see that this is, 1/1000th of that. So we're dealing with things that are very small. But we can pack these little systems with drug, and then the objective, of course, is, well, let's get it more specifically to a tumor. Now, just to orient, these are things that are real drivers, right? I mean, you've all had relatives who had issues with cancer, I'm sure. Right now, what we do is we give them a drug that goes everywhere in their body. And so less than 0.01% actually gets to the cancer. The rest of it goes through the rest of their body and makes their hair fall out, throw up, and all the rest of it. And so there's a real need to develop some delivery systems that just take the drug to where it's needed and spare the rest of your body. So this is these are the kinds of things that, very basic studies in membrane lipids, but very quickly you can get into things that have real therapeutic benefit. So this is me back in about 1990 or so. We started a company to use these liposomal systems to deliver cancer drugs to tumors. This is me and a few of my coworkers in the lab. I should say this is a great way to keep a team together. We're still working together one way or another about 40 years later. Tom Madden, for example, is the CEO of the company that actually has done the lipid nanoparticle work with the BioNTech in Germany. And so we started working on these cancer drugs, but very soon we started working on what we might term gene therapies. This is Jim Miller, who we hired as our CEO for INEX, and one day in about 1995, he came to me and he said, "I can't raise any money putting these cancer drugs into liposomes. We got to be doing gene therapy. That's much more sex appeal." That's exactly what he said. So from an investment point of view, that's what we started to do. So this was really gets us into the next topic, as it were. What is gene therapy? If you look at Wikipedia, you're going to find a pretty general definition that gene therapy is a therapeutic delivery of nucleic acid. It can be RNA or DNA, and I'm sure you've heard of nucleic acids in your biology courses, into a patient's cell as a drug to treat disease. And so what you want to do is get these big molecules, these big negatively charged molecules, into a cell in order to do gene therapy. Now, going way back in the history of nucleic acids, in 1953, we discovered this double helix structure for DNA, and it was clear that the sequences of the various bases in the DNA were coding for proteins in your body. He got a Nobel Prize in 1962, and then in 1961, it was discovered that DNA codes for proteins via a messenger RNA intermediate. And so there's transcription, messenger RNA is then transcribed in a ribosome to make a protein. This is what's termed a central dogma of molecular biology. So this is going on with amazing rapidity in your body as we speak. So there's a number of things that happened since 1961 that in the end led to the COVID-19 vaccines. Discovered mRNA. If we make mRNA and inject it into a cell, it makes a protein. The vaccines were tried, but modified RNA was shown to be non-toxic, so that was a big advance in 2005. In 2014, we showed that we can produce protein in the liver using lipid nanoparticles to deliver this messenger RNA, and it eventually culminated in the vaccine, as I've said. So as I said, in 2005, Drew Weissman there at the University of Pennsylvania, they discovered that how to make messenger RNA, that's this long piece of RNA that codes for a protein, they can make it less termed immunogenic, but in other words, really just much less toxic, and they could make it produce more copies of a protein by altering the chemistry. And so this is, they're co-winners of the Gairdner Award along with me. So it's, I've played a big role in this field. And then in 1998, a couple of investigators called Fire, and they're at Harvard, discovered that short pieces of RNA can control gene expression by binding to messenger RNA. So in other words, you could take, I'm going to go backwards here. You could take these are kind of short pieces of RNA. You could take these, and if you design them properly, they would bind to the messenger RNA and cause it to be degraded very, very specifically. And so this is a way of, if you want to term it, gene silencing. So they can basically, if you design them properly, they'll knock out, they'll silence any protein you want to silence in the cell. And so this really sets the stage for RNA molecules to be used as therapeutics. This is Fire and Mello. They got the Nobel Prize in 2006 for designing these short interfering RNAs. So these are potential therapeutics. So just think about it. With siRNA, you can silence any protein you want. So most cancers are associated with, say, an oncogene a protein that causes cells to divide rapidly. Well, let's just silence it, and in other words, we stop the production, and then maybe you can cure that cancer. Or on the other hand, for messenger RNA, you want to produce any protein you want. Say somebody is born as a hemophiliac, their blood doesn't clot, maybe they need a particular protein. Well, let's just have them make that protein, and then you're going to cure that disorder. So the therapeutic potential is huge, but they all require this delivery because they can't get into cells on their own. And so this is the point here. Using short interfering RNA or messenger RNA, we can potentially treat most diseases. And so that's kind of a big statement. But so you can see how it goes. I mean, that's what we try to do with most medicines, is try to cause one protein or another to be either to stop being effective or to start being effective. So now we're into a different problem. They both require these delivery systems. And so this is, the statement that so we can potentially cure most diseases. I'm not going to go through all of this slide, but I'll just say everything from heart disease to cancer to COVID-19 as a vaccine, strokes, respiratory diseases, Alzheimer's, you name it. All of those disorders, we can conceptually, at the very least, say, okay, these RNA approaches can be used to treat these disorders. Rare diseases, I mean, for example, that's 20% of all diseases, 35% of deaths in the first year of life. If a kid isn't making a clotting protein, well, let's get their liver to make it, and we can do that kind of thing now. So as I said, these are big molecules. They can't get into cells on their own, so they need delivery systems to be effective. And so that's what we decided to do, or try to do, in about 1995. It's a pretty big challenge because you have an immune system, and one thing your immune system wants to do is to stop any foreign genetic information getting into your cells. And so the idea of packaging up a nucleic acid and then injecting it intravenously and having it go into your body, into a particular tissue, and then into a cell, and then get into the interior of that cell, well, this is a tough problem. So we hit a major problem right off the bat, and that was that the RNA, DNA polymers, they're really highly negatively charged. And if we want to get them into a lipid nanoparticle or something that is hydrophobic, we had to use these positively charged lipids, cationic lipids, in order to do that. Now, they're really toxic molecules. So in the end, we knew we couldn't do it with using what was available at that point. But what we had was these ionizable cationic lipids that I mentioned for their lipid asymmetry work. So we thought, okay, well, maybe we can encapsulate at the low pH, where they're positively charged, encapsulate these nucleic acids, and maybe they'll be retained in the lipid nanoparticle when we raise the pH back up. As I mentioned, this is about the acidity of a lemon, so it's not that acid. We bring it back up to physiological pH. It turned out to work really remarkably well. So this is another piece of serendipity in a way. After a conference I was at in 2004, I was pursued by, I always characterized him as a mad Russian. He was a bit mad, but anyway, he was a VP research, vice president research for a company in Boston called Alnylam. And they were founded to deliver the siRNA to the liver to silence a pathological protein. They said, why do we have a delivery problem? How do we get our RNA into the liver, into particular cells in the liver called hepatocytes in vivo? And so we set up a collaboration between my lab at UBC and Acuitas to do just that. We worked together for about 7 years. Some of these collaborations, we were synthesizing many different things and testing many different formulations. And this resulted in a formulation where we could encapsulate in these lipid nanoparticles siRNA, and then they could actually silence a gene in the liver. This is where we get quite technical. We used an assay where we were basically injecting lipid nanoparticles containing an siRNA to silence a marker protein called factor 7, which is a clotting protein. And using this, we could optimize our system. We increased the potency by 3 or 4 orders of magnitude. And the design of these lipids actually relied on our understanding of lipid polymorphism, which is one of the basic things that I was mentioning at the beginning. So basic research gives you tools and insights that you wouldn't otherwise have. Anyway, so we went on to say, okay, well, what disease should we try and treat? And the clinicians at Alnylam said, okay, well, we should go after this disease you've never heard of called transthyretin-induced amyloidosis. a pretty nasty disease, actually. Transthyretin is a protein that's made in the liver, expressed in the liver. It's a transport protein in the circulation, but if there's mutations in it, it can form these long fibrils, and these fibrils will deposit, all the way through your body. But they have really nasty effects if it happens in your nerve tissue or in your heart. And so these, after diagnosis, you basically have about 5 years to live. So this is a kind of a progression that you can see these patients go through over this 5-year period. It's a wasting disorder, and there's currently no treatment. So you can imagine how bad this feels for people that are diagnosed with this. They have trouble walking in their 30s, and they probably have a relative that's gone through the process, so they would see what they were in for. Anyway, so simple solution here, let's just stop that protein being made. So we put this sRNA to silence that particular protein in our lipid nanoparticles, and it's potentially a simple solution in what is obviously a devastating disease. Let's go on here. There was a clinical trial done that finished about 2017. This was an international trial. Things like neural impairment score, their quality of life, self-reported, weakness, et cetera, were the endpoints. But basically what was shown over the 18-month course was that these patients were actually, if they had a placebo, which is just sterile saline, they got worse, as according to the neural impairment score, whereas if they had the drug that was silencing that particular protein in their liver, then they were getting, if anything, getting better. And so we're taking a hereditary disease and essentially curing them of it. So this is just the clinical trial results were absolutely amazing from all points of view. So this is a curative therapy for what was previously a fatal disorder. This is some of the things you can do if you get into the life sciences, and obviously we were particularly fortunate, but it's amazing the impacts that one can have on people's lives. So this was approved by the FDA, the U.S. FDA, in 2018, and it really dramatically demonstrates the power of these kinds of gene therapies. It's fairly simple to decide what your approach should be, and using these big molecules, and it certainly is very powerful. So we then asked the question, well, if we can deliver this small interfering RNA to the liver to silence a gene, can we also deliver messenger RNA to express a gene, in other words, to make a protein in the liver? And this turned out to be doable. You may be aware your liver makes about 70% of the proteins in your body. It's a protein factory. And so we can actually ask it to make some more proteins of any that we want just by putting the messenger RNA in our lipid nanoparticle and then injecting it into the person, goes to their liver, the hepatocytes start to make that protein. This is just, you might have heard of erythropoietin being used by cyclists in the Tour de France. This is where we're taking it, this is in pigs. We can get supramolecular levels of erythropoietin in the circulation of these animals just by injecting these. So you can really start to change around the biology in a fascinating manner. But we were approached by Drew Weissman, as I mentioned, and Katie Carrico, who are co-winners of the Gairdner Award with me. This is around about 2014, saying, okay, you've got things that work pretty well in the liver for making proteins. Can we try them to see if they'll also be useful as vaccines? So this is one of these things where, this is the zigzag approach in science. Unexpected things can occur. So they had a delivery problem. How do we get mRNA coding for, say, a viral protein in the case of a vaccine? How can we get that into muscle and immune cells in vivo? So they wanted a different thing. They wanted something where they're injecting intramuscularly, and then wanted to get an immune response resulting from expression of the viral proteins in antigen-presenting cells and also in the muscle cells themselves. This worked incredibly well. The first thing that we looked at was Zika virus. And so the Zika virus, we put into a lipid nanoparticle, we put mRNA coding for one of the proteins that's on the surface of the Zika virus, and injected that into animals, and and then challenged them at 2 weeks or 20 weeks with lethal doses of the Zika virus itself. And what we saw was that they provided total protection. In other words, they didn't get infected. So this was a very, very good vaccine. And so this led to Acuitas, which is one of the companies I co-founded, to work with BioNTech to develop an influenza vaccine. This was around about 2018. And BioNTech was also, this is a company in Germany, was also working with Pfizer on a flu vaccine. Well, in early 2020, when the pandemic hit, all the efforts switched to making a COVID-19 vaccine. So this is in February of 2020. And so here, in this case, we're using the same lipid nanoparticle that we're using for the liver applications. But now we're putting in messenger RNA coding for the spike glycoprotein, SARS-CoV-2 spike glycoprotein, and putting the messenger RNA that codes for that. And all of you know the results that resulted from that, where we're seeing 95% effective. This vaccine is 95% effective for preventing infection of with the SARS-CoV-2 virus, with preventing COVID-19. This was effective across all age groups and was reasonably well tolerated. They said they were going to produce, I think, 1.3 billion doses by the end of 2021. There were closer to 3 billion doses that were produced, and of course it's been approved all over the place. So, but really these vaccine applications are just the tip of the iceberg. There's many, many more, obviously, vaccines that we can think of HIV, Zika, of course, but for chronic diseases, for treatment of cancer, heart disease, Alzheimer's, all of these diseases now, there's potential therapeutics that are on the table. For example, if we inject these things into the brain, we can express any protein we want, and there's huge opportunities there. Into the eye for eye diseases, inherited disorders, rare diseases, the list goes on and on. So it's a very exciting time in medicine when we suddenly can use a whole new raft of potential therapeutics. And so I hope I've explained to you how life science research, basic research, can be pretty interesting. You're trying to understand how nature works. And I got interested in lipids, but really once you understand how things work, the opportunities for developing new drugs and new ways to approach disease really become much more apparent. And in my case, obviously we've developed quite a number. So with that, I'll close, but there's a very exciting time in medicine at the moment using these kinds of approaches. I hope I managed to convince you of that. So thank you very much. Yeah, so I'll take any questions anybody may have. Yeah, if you can come to the mic, or otherwise I'll repeat your question if I hear it. But, this is always the part where, who's going to dare to get up and say something? But okay.
Does the COVID pill that was also developed also use the same technology that you had researched? Yeah,
so the question was, does the COVID pills also developed use the same technology? The answer is no. No, that's using a different approach to basically decrease the infectiveness of the virus itself. So it's not a vaccine. Yes? I mean, you can just yell from... What, pardon me? Have we made any vaccines? Well, some of the, obviously the vaccine that we're producing for the, for COVID-19 is an example of a vaccine. I think the ones, oh dear, the ones that are going to come along in the next while, it's most likely going to be for Zika virus, influenza. You're going to see a universal flu vaccine, so you don't have to go in every year to get a, and that'd be much more effective to get a new shot. So these are vaccines against cancer, I think, are going to be, the idea there is you just take if somebody has cancer, then you pull out some of the cancerous tissue, identify the proteins that are associated with that tissue, and then make a vaccine that makes the body have a reaction to those proteins. So these are things you're going to see. The new vaccines are going to be very, they're very rapidly developed. We can develop in a time on the order of a few weeks, and you have some highly personalized vaccines that'll very likely work well on you. Yep.
Are there any drawbacks to this technique, or is it just all good?
Well, it's like, I mean, for example, HIV vaccine, there's huge efforts going on to. The question was, are there any drawbacks? And there's always drawbacks. As you know, there's some people have a bit of a reaction to the COVID-19 vaccines. But the basic answer is that they're a big advance on what we've got now. We have to design them so that the immune response lasts longer. Maybe we want to be able to decrease the dose so we don't see any side effects. So those are things that are under active research now. But they look like an amazing advance on previous technology for vaccines, that's for sure, but also for other therapeutics. Yep.
I just had a question.
Yeah.
So, in your opinion, what are better, vaccines or taking pills for, like, flus?
So the question is?
In your opinion, what do you think are better, taking vaccines or like pills for flus, or like?
Oh, it's the pills to boost your immune system or something. Well, I mean, vaccines are probably saved more lives than any other single form of medicine. So, I mean, a vaccine is a pretty good, invention. As we take more and more pills, particularly small molecules, so they have all sorts of other side effects, which can have real issues. So, I think if you're preventing yourself from getting a disease as opposed to treating it after you've got it, it's it's a better way to go. Yep. Will there be more what? Oh yeah. No, that's a big advantage of this approach, is that you can make a new vaccine. Well, you saw it happen with the COVID-19 vaccines, right? I mean, 3 months after the SARS-CoV-2 virus, COVID-19 became apparent, they were in clinical trials with the vaccine. That was because they can just, once you know what the sequence of the protein is, you can make a messenger RNA, encapsulate that in the lipid nanoparticle. You can do that very fast, and so within a month or so. And so they're much faster and they're much cheaper. And you can actually, and it's a bunch of advantages. The manufacturing process is much simpler. Yep. There's two of you. Okay. Whoever wants to speak first. Oh, you're down there. Okay.
mRNA vaccines being like a new technique, has there been research into more of the long-term effects?
Well, just about as long as we've had the COVID-19 vaccine, right? I mean, this is so it's pretty I mean, when was the results announced? It was November 2020. So from a long-term point of view, no, there hasn't been, you know, I mean, we're gradually gathering more and more data now. We're finding that perhaps you know, some of the immune responses don't last as long as we might like them, and so and then the booster things for various variants of the disease. And some people have, you know, we see some small cohort, 1 to 2%, that might have a reaction to some of the components, but that's typical for most vaccines, that they have, you know, there will be a small cohort that will have, you know, and we've grown quite expert at controlling that in terms of, you know, any. So I don't think, you know, the evidence so far would say that it's, you know, the benefits are extreme, you know, look good, and the downside is not so, you know, it's not so apparent. Now, that's not to say there may not be something come along, but one thing that's true about these kind of drugs, you know, I refer to them as kind of gene therapies, but they're not affecting the genome itself. They're not affecting the DNA inside the cells. They're only operating outside the nucleus in the cytoplasm. And so, in that sense, they're very safe. You know, we're not affecting your DNA, and I mean, the things that might actually go on and have some long-term effects. Yep. Yeah. How widespread? It's getting pretty widespread. You know, there must be. In the Boston area, for example, I think there must be at least 20 new companies that are, you know, and this is all happening all over actually, that are developing new therapeutics based on mRNA and lipid nanoparticles. They're treating everything from heart disease to atherosclerosis to cancer. You know, there's been literally billions of dollars of investment gone in the last couple of years to start to develop these drugs. So you're going to see a lot of them coming onto the market in you know, over the next few years. The evidence so far is that, you know, these things are remarkably effective. So it's a bit of a revolution in therapeutics, and it's having certainly the amount of investment going in, and the excitement is really noticeable. Yep.
From the 3 drugs that were given to treat cancer, which one is the most successful with these
So you're asking the question, from the drugs which are used to treat cancer, which are the most successful? With a new technique? Yeah, there's nothing yet in the clinic to treat cancer using these approaches. Well, there's some things that are in the clinic, but they're not approved. And so I guess the lead one would be something called CAR T-cell therapy. You know, that's one of the lead ones. But that one's very exciting, because what it does, what the approach there is to essentially take T cells, which are kind of killer cells in your immune system, and target them more specifically to cancer cells. And so the evidence, it's looking like we're going to be able to reprogram T cells using these lipid nanoparticle approaches to specifically go after particular forms, particular cancers. And so that's one that's the cancer vaccines is another one that is really receiving a lot of emphasis. So companies like BioNTech and Moderna and others are really pursuing that kind of thing very hard. Yeah? Oh, sorry, you're very faint. Have I had any complete failures? Oh, many. Yeah, I wouldn't say they're complete failures, but you learn something from them. You know, we've done a lot of work with taking normal anti-cancer drugs and trying to get them more specifically to tumors. And you know, that, which, as I was pointing out, is really something we need to do. You know, because right now, with these drugs have really nasty side effects. And we did succeed to some extent. So we developed a drug. You know, it was one of these, exactly these kinds of systems. And we invested a huge amount of effort and money. And then in 2004, we took it to the US FDA to see if they would grant us approval, and we didn't get it. And you know, they said you go back and do more studies because they weren't convinced. And so yeah, that was you know, because we raised almost $100 million in investment to take the drug through clinical trials and all the rest of it. And so that was a pretty big, the drug eventually did get approved, but we had to license it out. We had no more money to continue the development. And so, yeah, that was not a good moment in my life, that's for sure. Sorry, speak a bit louder. Oh, yeah, no, all types of cancer are open. Yeah, it's really interesting with cancer. I mean, if you think about it, they all end up being. Oh, sorry, if you go ask some more. Yeah, well, the vaccine approach, for example, you're not going to have the toxicities that are normally associated with chemotherapy. If we can deliver cancer drugs more specifically to a tumor, we'll avoid those toxicities as well. So yeah, I think these approaches are going to be curative for cancer. We just have to get them working. Yep. How are they made? So the question is, how are these lipid nanoparticles made? Well, this is one of the reasons to use lipids. That have this, they're self-assembling. So what we do is we just mix them together. You know, we have the lipid that's dissolved in ethanol, and we have the mRNA or the siRNA dissolved in water at pH 4. And we mix them together, and that's basically it. If we put in the right components, then they will self-assemble into these, you know, 100 nanometer or a bit smaller nanoparticles. And so we're taking advantage of, you know, properties of membrane lipids that are really very useful. So you don't have to sort of manufacture it and say, okay, let's put this bit in and that bit in. They self-assemble into these finished structures. It's a big advantage of the approach. Yeah. Sorry, vaccines for what? Sorry, which disease? Oh, diabetes. Vaccines for diabetes. No. We're working on other areas in diabetes, but not vaccines for diabetes, no. I'm just trying to think of how one might actually do that. It might be kind of tricky. Not every disease can you approach using a vaccine approach. Yep. What are the favorite things? The speed, I guess. You know, I mean, once you say you want to go after a particular infectious disease, you can come up with a new vaccine in a very, very short time period. I mean, a matter of a month or two. So it's that's and they're very, very potent. You know, we're going to see these systems become much more potent too. We haven't really, you know, optimized them yet. We've optimized them for the liver, but we haven't really optimized them for vaccine applications. So, yeah, it's you know, in the beginning of a field like this, it's it's kind of a fun time because there's lots of different approaches to take, all of which could potentially improve the properties. Yep.
So there was like a little graph thing with various methods that your RNA and lipid nanoparticles could help solve common conditions.
Mm-hmm.
But for the COVID section, it just said helps combat COVID-19 or fight COVID. How exactly does it help fight COVID if it's not too common?
You mean as a vaccine? I mean, what it's doing is it's, you know, so you're saying, okay, the COVID-19 is caused by a virus called SARS-CoV-2. And associated with that virus, there's a membrane protein called the spike protein, spike glycoprotein. And what you do is you make the mRNA that codes for that particular protein. And then if that gets made inside a cell in your body, then your body will say this is a foreign protein and raise an immune response to it. Because it has an immune response to the spike glycoprotein, it'll then recognize the SARS-CoV-2 virus itself because that has the same protein associated with it. And so the immune system is then primed to go after the virus if it sees it in your body. Yes, I was at the end. Not you.
So, we talk about cancer drugs. It can go everywhere in the body and can cause some nasty side effects. What kind of effects?
Oh, your hair falling out, throwing up, heart failure, can cause cancer themselves. You name it. I mean, these are nasty drugs. So, yeah, the ability to focus them into you know, the area where they're really needed and not have them go elsewhere in your body is really an important one to get over. Yeah.
So, do they convert from generation to generation?
So the second generation of what?
Like, if they have some nasty side effects, the side effects will be transferred from generation to generation.
I don't know if I quite understand the question. You said,
If they have vaccination and someone have faced some nasty side effect of cancer drugs, so that will be traveled to like his son or grandson.
oh no. It's not something that you carry. It's just for that one individual. It's not like they're. They may be more susceptible to certain side effects because of other issues, but because you know one person gets it, or has a bad reaction to a drug, doesn't necessarily mean their offspring or relatives are going to have that. Yep.
How do you insert the mRNA into the LNP?
Yeah, that's the trick of the ionizable lipid, right? So the lipid at pH 4, it has a positive charge. The mRNA has a negative charge. And so when you mix them together, they stick together in these. It's like a little fatty bubble, if you want to think of it that way. So it's just this electrostatic reaction. It's like having two magnets come together, and they're going to stick together. So. Okay, so it looks like there's no more questions. So, thank you.